Yeah, so we've got 3 minutes or so. Let people keep on coming in.
Yeah.
Finish my
Okay.
coffee.
So Johnny's from Brisbane, Amy's from Hong Kong. Can everyone else share where you are watching this from?
Oh, nutrigenomics. That's some interesting stuff, Amy.
Yeah, it's hitting Asia. Okay. William's from Auburn, Alabama. Okay, so you're working on your own metabolic problems. So, hi everyone, my name is Nata. So if you're a reader of SelfHack as well, I'm the chief editor of SelfHack. So today I'll be the co-host. I'm helping out with Brendan doing this presentation to share with you all about genetics and science behind SelfDecode. Okay, cool. We're just gonna wait a few more minutes before we get started. So. We have a few very interesting questions here. So you guys are pretty intelligent. So Johnny said treating SNPs, methylation issues, and mental health field. Okay, methylation is a pretty popular topic now.
Yeah, I mean, methylation of genes, you mean epigenetics?
Yes, there's methylation of genes, but like the SNPs that they usually talk about is the MTHFR SNPs, right? And it can methylate a few things. It can methylate hormones, some enzymes. Like it has a few different substrates other than their DNA, but DNA is one of the substrates as well.
Okay, cool.
Yeah, it's definitely one of the topics that's not commonly misunderstood, I would say.
Yeah, I mean, epigenetics isn't, it's advanced genetics. It's not genetics 101. I'd be really keen to do a future webinar covering epigenetics in more detail.
Cool. Yeah, that should be fun, and especially if we can make it accessible for our users.
Yeah.
All right. Okay. Well. Okay. So it's like 1 minute too. I'm just gonna start introducing you, and I guess a few more people will show up as we go along. Right. So it's a pleasure to introduce Dr. Brendan Swan. He has a PhD in neurogenetics from the University of Auckland in New Zealand. Right?
Yep.
And he did the studies looking at identifying mutations that cause complex neurodevelopmental disorders like autism. And then he looked at the causative mechanism. So how exactly these mutations cause their complex diseases like autism? And the reason that we chose Brendan to join our team is this is because a lot of people who use SelfDecode have a complex health issues, and we are trying to figure out what causes them genetically, right? So to Brendan, SelfDecode represents the future of health and well-being, and it's a tool that provides personalized information about an individual's genetic variations. So it's a place for you to learn how to align your lifestyle with your genetic makeup in order to have longer, healthier, and happier lives. Okay. So now Amy asked about genes associated with autism now. All right, great. So we'll address that after their presentation. And I've seen this presentation in the rehearsal, and I know that Bernard is an amazing instructor. So, all right.
Cool. Thanks, Natha. Natha, can you keep an eye on the questions and just keep a record of them, because I don't want to lose them as they go up the chat.
No, if I can answer it, I will answer it in the chat box, because I know I have an accent, but I also have a PhD in molecular genetics. So I'll try my best to address as many questions as possible, and then hand over their more complex ones to you. Okay?
Okay. All right. Oh, good luck, both of us. All right, I'm just going to get started then. Okay, greetings everyone and welcome to this webinar. Okay, today my main goal is to cover some of the basics of genetics to give you a better understanding of what SelfDecode offers and how to get the most out of it. Okay, I'm going to start with the absolute basics and then build up from there. So for those of you who already know the basics of genetics, just please bear with me for a while. And for those of you who are new to genetics and feel like you get a bit lost, don't worry, we'll have some time at the end for any questions you might have. So I'll just do a brief overview of how I want to do this. I'm going to do the first half as a presentation. So it'll be me talking and going through some information. And then I'll give some demonstrations with SelfDecode about how to use that information. And then we'll finish with some question times. So just quickly, I want to cover the basic building blocks of DNA, how we arrange these into genes, the process by which genes are turned into proteins. That is how information stored in genes becomes a three-dimensional functional thing whizzing around the cell. Then I want to move back to the DNA sequences and talk about genetic variation, specifically the SNPs that everyone's so interested in. We'll cover the meaning of major and minor alleles and look briefly at how a risk allele is identified. And I want to give you some tips about how to interpret risk alleles, so to understand the statistics and how scientists are identifying them. Hopefully this will give you some insight into how you can use SelfDecode.
Hey, Daddy.
What?
You took off that light so I didn't know you.
Okay, sorry about that. And I'll give you some insight into how you can use SelfDecode to gain information to make yourself healthier and happier too. All right, so I'll just get started. So genetics 101. So the best place to start is at the bottom. The base of genetics are the bases. Those four letters you hear so often, A, T, G, and C. The nitrogenous bases, don't worry about the chemical structure if you find it confusing. Just focus on their colors. There won't be a test at the end of this, so don't worry. But bases by themselves aren't enough. You've got to add a sugar and a phosphate group to each one. This makes a nucleotide. These are the building blocks of DNA. So your sugar is a ribose sugar, 5-carbon sugar, and your phosphate group beside it. DNA stands for deoxyribose nucleic acid, and that ribose is the sugar, joining the phosphate and the base. Okay. But the DNA that interests us are the long sequences found within our cells. Those building blocks are attached together, with the phosphate and ribose linking them into a long chain. These chains can be millions of nucleotides long or bases long. So a more simplified representation of this chain is here. You can see the phosphate and ribose backbone and a short chain of nucleotides. You've got your A, your T, your G, and your C. And again here, A, T, G, and C, just represented more simply. Now, one chain of DNA has the ability to then bond to another chain with complementary bases. So complementary bases are A with T, T with A, G with C, and C with G. It's always this way. And the reason is A and T have 2 hydrogen bonds, these little dotted lines, while G and C have 3, these little dotted lines. So they preferentially bind with each other, and that makes a double-stranded DNA. Well, that classic DNA double-stranded helix is derived from these, combining all these nucleotides together, making 2 big long chains that then twist into your DNA double helix. Okay, so now we've got something to work with. We can start gaining a better understanding of just exactly what our genetic makeup is and how it contributes to who we are. This will help us to get the most out of SelfDecode. Right, our bodies contain a lot of DNA. There are about 6 billion nucleotides in the average cell. That's enough DNA to stretch about 6 feet long. Now, if there are 30 trillion cells in the human body, that means in a single typical adult, there's enough DNA to stretch to Pluto and back. Why do we need so much? Why do we go to all the effort of making so much DNA? Well, our DNA contains all the information required to create and sustain us. We inherit half of what we have from our mother and the other half from our father. So let's start with that very first cell, a fertilized egg, the zygote. We were all once a single tiny cell, and thanks to the genetic information stored within, we were able to grow into the people we are today. So to start with, let's look at where all the DNA is kept in a cell. This is the nucleus. It's like a sphere within the sphere of the cell. Our DNA in its double helix form is then wound up in these super tight, very complex structures called chromosomes. Now, if we zoom in on the nucleus, we can see that there's lots of little chromosomes in here. So in a typical cell, we have 46 chromosomes. 23 came from mum and 23 came from dad. The XY and XX chromosomes are what differentiate between males and females. This constitutes our genome. We have the same genome as the first cell in every other cell of our body. So that first zygote has the same sequence of 6 billion bases as every cell in our body. Our genes are encoded within the chromosomes. Okay, the next thing we need to cover is what a gene is and how genes go about helping that single cell, that tiny little zygote, grow into an adult human. A gene is a sequence of DNA that encodes for a protein. This is a very important concept, and I'm going to go over it in a bit of detail. Genes encode information that is vital to the development and maintenance of cells, and thus us. But how does that information get used? A key part is turning genes on and off. Turning on a gene is referred to as promoting, activating, or expressing it. Turning off a gene is referred to as repressing, deactivating, or suppressing. Controlling when genes are turned on and off is called genetic regulation, or another word for it is epigenetics, like we've been talking about. So what happens when a gene is turned on? The gene sequence, say here, up here, is transcribed from DNA into RNA. Then the sequence is translated from RNA into a chain of amino acids. These form a protein. Essentially, two steps: transcription and translation. Okay, a gene is a sequence of DNA that is copied into RNA. RNA is another nucleic acid like DNA, but slightly less stable. This process is called transcription because the sequence is transcribed. The A's go to A's, the G's go to G's, the C's go to C's, and the T's become U's. The RNA T is a U, it's just the way it is. Okay, the RNA is a single strand of nucleotides. So DNA is often double-stranded, RNA is single-stranded. This is called the messenger RNA because it carries the message of the gene. The RNA copy of the gene is transported out of the nucleus, so that bit inside the cell where all the DNA is, to the protein-making factory of the cell, also known as the endoplasmic reticulum, which is one of my favorite words in biology. At the protein-making factory, the gene sequence is encoded in the RNA is used as a template to create a protein. This step here is known as translation. The sequence of the gene is translated from the RNA into the amino acids that make up a protein. So this next bit is pretty essential, so just pay careful attention. It's a bit weird. You've got groups of 3 nucleotides. So here you have AUG and CUU. These 3 nucleotides, they are used to code for specific amino acids. In this case, AUG codes for methionine, the amino acid methionine. CUU codes for the amino acid leucine. UCG codes for the amino acid serine. And UAU codes for the amino acid tyrosine. As the sequence of the gene is read, as the sequence of the gene is read, every 3 bases an amino acid is added to a lengthening chain of amino acids. So as you go through the sequence, you get more and more amino acids being added together, and a big long chain of them is formed. This is what a protein is. Proteins are made up of chains of amino acids. They can be 100 or so long, or they can be tens of thousands amino acids long. And there are 20 commonly used amino acids to make proteins, and each common amino acid has a specific trio of nucleotides that codes for it. These 3 RNA nucleotides are called codons. So this table shows you the codons, so GCU or UGC, that code for different amino acids. Don't worry about this too much, don't worry about the details. I just want you to know that the sequence of the RNA or the DNA of the gene is then translated into amino acids. And you've also got a start, which tells the protein making factory to start adding amino acids together, and you've got a stop codon, or 3 stop codons, that tells the protein making factory that that's enough, and this big long chain of amino acids can now be released into the cell. What I want to get across here is how the complexity increases as we transfer from DNA to amino acids. How those 4 bases encode 20 amino acids. Okay. Don't worry about this slide too much. We won't linger on it. I just want to reinforce how we go from the sequence of a gene, a 2-dimensional sequence, into the creation of a 3-dimensional functional protein. This is an essential part of biology, especially genetics. So amino acids essentially have the same basic structure. You've got a central carbon with 3 arms sticking out. So you've got this NH2, you've got the amine group, and you've got the carboxyl group. So all of them have this bit in the middle with the same black. For the black writing, it's the same. You have this bit in the middle and then 2 sticking out. Bit in the middle, 2 sticking out. Bit in the middle, 2 sticking out. You can also see each amino acid has a different bit, the red bit sticking out the side of it. Now the red bit is what makes each amino acid functionally different. And this is referred to as the R group. And they vary in size and chemical properties. Like some are acidic, some are basic, some are non-polar, and some are polar. So a non-polar amino acid doesn't like being in water, while a polar amino acid does like being in water. This is important because cells are basically full of water. Okay, I'll move on to the next step. So the amine and carboxyl arms are used to join the amino acids together into long chains. So you've got this little bond here, it happens again, and you just keep adding them, and you get a chain of amino acids with different R groups sticking out. As all the different chemical properties of the R groups, they interact with each other and their environment in different ways, it causes the chain to fold and bend. Some might be attracted, some might be repulsed. And this creates the 3-dimensional structures of proteins. So in this image, the white line is a chain of amino acids. And then you've got R groups. You've got polar R groups and non-polar R groups. So the polar ones want to be in the water. The non-polar want to be away from the water. So what happens is that chain will then fold into a 3-dimensional shape where all the polar amino acids are on the outside and all the non-polar are on the inside. This creates a structure known as a protein. And a protein can be structural. It can be like collagen, which is in your skin and your hair and everywhere, which helps make cells strong. And it can be functional, like an enzyme that facilitates biomolecular reaction. This turns the information stored in the sequence of a gene, or that's DNA in your nucleus, into a thing that can go out into the cell and do stuff and change and make a difference in your cell and in the way you react with your environment. Like I said, proteins can be structural, they can be enzymes that facilitate chemical reactions. They can also be controlling units that turn other bits on and off. Proteins facilitate growth, metabolism, defense, pretty much anything else a cell does. Okay, we'll move away from the proteins now. We'll move back to the genetics. This is Genetics 101. But I really wanted to reinforce the fact that genes are encoding for proteins, and proteins are what create us, our phenotypes, our different traits. So if we bring our story back to that tiny single cell, that zygote that we all once were, using the genetic information received from our parents, we began to do things in that cell. Genes were used to create proteins, which help the cell to grow, to process nutrients and energy, to create tools that copy DNA and other cellular parts, and allow the cell to duplicate. So that first cell became 2, and then 4, and then 8, and so on. This process has happened trillions of times. We formed different organs, we formed neurons and brains, and slowly we became the people we are today. So for those of you who don't know me, that's what I look like. All of this is facilitated by the information stored in our DNA sequence, our genes. So understanding how our genes work and what their specific sequence is can give us valuable insights to how our bodies and minds work and allow for us to use this information to create healthier and happier lives for ourselves. Our goal at SelfDecode is to help you realize this. It's complicated and we're working hard to make it less complicated and easier for you to understand. Okay, so I'll just quickly recap that little introduction and then I'll move on to focusing on SNPs and how SNPs fall into it. But I really wanted to just quickly cover how DNA becomes proteins which help us create. So we introduced the building blocks of DNA, the nucleotides A, T, G, and C. These join together to form long DNA strands. Complementary strands bind together and twist together into the double helix shape. Genes are special sequences that get copied into RNA, which is then used as a template to translate the code into a chain of amino acids. These chains of amino acids will then fold into 3D, fully functional structures that can carry out a task within the cell. Okay, moving along. Now we want to focus on the sequence of DNA and how the sequence can have different variations and what this might mean for you. There are several types of genetic variation, but we're going to focus on a single nucleotide variation, SNV. So in this example, we're replacing a G with an A. That is a single nucleotide variation. The G became an A. Now, if only a very small number of people have an A, then we might call this a mutation. If it's more commonly observed, then it would be referred to as a single nucleotide polymorphism, or SNP. So these genetic variations are what we focus on at SelfDecode. Differences in the DNA sequence between you and the typical sequence. And they provide clues to traits you might be susceptible to and provide ways of ameliorating or treating those things. SNPs are typically named with the letters R and S, which stands for reference SNP, and followed by a unique number. The majority of your SNPs will not be found within a gene sequence, but I want to quickly talk about what can happen when they are, just to give you an idea of what nucleotide variations can do. Okay, as we saw, the sequence encoded in a gene is essential for the formation of its protein. So in this case, we have a start codon, resulting in a chain of amino acids, and a stop codon. This creates a structural protein. But changes to the sequence result in different proteins. So we've got three types of changes I'll quickly cover. The first, if this G becomes a T, so instead of CGG, we have CGT, this is known as a synonymous or silent mutation because CGG and CGT both code for arginine. So there's been a variation, but there's no change to the amino acids. The next one I want to talk about is changing, say, this A to a T, so AGC to TGC. Now, TGC codes for glycine, not serine, so we've now changed an amino acid. This is called non-synonymous or missense variant. It might not change the overall structure of the protein, but it might give it a new property, like a new color. And the final one, if we change this G to a T, we get a premature stop codon. So now, this is called a stop gain, a truncation, or a nonsense protein, and we get a whole new shape to the protein, and often it's not a good one. So this a quick introduction to types of single nucleotide variations. Some of our SNPs are these that we can have. But for now, it's sufficient for you to know that some SNPs may have an impact on genes, but it's also important to know that many SNPs don't have an impact on genes. SelfDecode has information pages on tens of thousands of SNPs, so you can go along and check out information on particular SNPs and learn a bit more about ones you're interested in. Okay, now I want to quickly cover how we represent our SNPs. When that first cell, that zygote that would later become you, was first formed, it inherited genetic material from your mum and some from your dad. So for every gene, we have a copy from mum and a copy from dad. So for every position in the genome, we have 2 possibilities. In this case, just looking at the third base, the one with the red arrow, we can see that mum and dad had a G, and thus this person's genotype is GG, which we all call homozygous reference or major. If the base is the same for both positions, then it's homozygous. If dad had a T instead of a G, then this would be called heterozygous. If mum had a T instead of a G, this would be a TT, and we'd call this person homozygous alternate or minor. So the question now becomes, how do we know what is major or common and what is minor or alternate? Now, to answer this question, we needed genetic information from lots of people. As more and more people have had their genetics tested, the genotypes have been anonymously submitted to databases. These databases then calculate the percentage of genotypes for a given position, and then they tell you. You go to their database and it'll give you information. So this is a very useful piece of information. It's called population frequency. It shows you how often a SNP is observed amongst a group of people. From our case before, say the GT SNP, we can see that 59% of the people are homozygous major, 33% are heterozygous, and 8% are homozygous minor for the TT allele. So the most common variant is called the major, while the rarer is called the minor. So when we're talking about alleles, we're actually just referring to the G or the T and not talking about genotypes. So in this case, the T allele makes up 8% plus half of the heterozygous cases, so 16.5. So 24.5% of all observations are T, and the remainder are G. So the minor allele frequency here would be 24.5% or 0.245. Okay. SelfDecode includes this information when available, but it's important to know that not all SNPs have population frequency data. When we can find it, we'll add it, but it's not always going to be available. For the vast majority of the positions in your genome, you'll be homozygous for the major allele, so over 99.9%. It's been estimated the average genome has about 4 million SNPs in the 6 billion positions. And you've got to take note that most of these won't fall within a gene. Maybe 20,000 or so, 50,000 will occur within genes. The vast majority of your SNPs will be in non-gene coding regions. It's an important message to take home that many of your SNPs will not have any known impact. 23andMe and Ancestry.com include plenty of these SNPs in their tests because they give them good information about your ancestry, but they don't tell you anything about health and well-being, so they're less useful to us. The question now becomes, how do we know what is a good allele, a neutral allele, and a bad or risk allele? This is a tricky question, and we've still got a long way to go to figure them all out. But we've made a pretty good start, and as we learn more information, we're going to share it with you as we go along. Now, our best resources to answer this question come from scientific research. The most commonly used study for identifying risk SNPs is the Genome-Wide Association Study, or GWAS. Okay, often, how this works, a study will take 2 groups of people, ideally with lots and lots of people, one with a trait and one without a trait. Say for this case, the group on the left have diabetes while the group on the right don't have diabetes. The next step is they will genotype them. Okay, so this is what 23andMe and Ancestry.com do. They will identify each individual SNP and record whether the person has it or not. This is done using a special chip known as a DNA microarray or a genotyping chip. Essentially, this is a glass slide with millions of synthesized DNA strands attached in a fixed order. Each DNA strand is the sequence surrounding a SNP. So to genotype someone, you isolate a sample of their DNA, say from their saliva, and then you wash it over the slide with some fluorescent dyes. So when the DNA on the chip binds with the person's DNA, so that is the complementary, the person has that sequence, has that SNP, the dye will also be incorporated and it will light up. You can look at the chip and you can record what SNPs that person has. So the genotyping chip currently used by 23andMe, that has 602,000 SNPs. So 602,000 SNPs embedded on the surface of the slide. Okay, but back to GWAS. So once everyone in the two groups are genotyped, then the scientists will then compare the two groups and look for SNPs that occur within one group but not the other. So in our example, we'd be looking for a SNP that all the people with diabetes have and all the people without diabetes don't carry that SNP. But in reality, it's never that clear cut, unfortunately. So the statistics behind defining what is a significant SNP and what isn't is pretty complicated. We're basically looking for SNPs that are more common in one group and less common in the other. But to get this statistical significance, there's a lot of background maths. It's important to remember that these studies are looking for association and not causation. So while the associated SNP might be there every time someone has the trait, it's linked to the genetic variation that causes the trait. It's not necessarily the genetic variation that is the reason for the variation or the trait. Identifying the causative SNPs requires a slightly different approach. And I'll just cover that quickly here. Just to reply to that 23andMe and Ancestry, they use different genotyping chips, sorry. 23andMe has one that they've made specifically for themselves for their targeted SNPs. So the sequencing studies, they're similar to GWAS. They involve two groups, one with a trait of interest, one without. But instead of looking at a selection of the common SNPs, they use DNA sequencing technology to sequence everything. So they're looking that they'll have information about all the common SNPs that are there, but they'll also identify rare variants that might not have, wouldn't be included in a SNP chip, and they'll be able to identify where you're the same. So in this picture here, say all the white is the same, and then the red is where you might differ, where there might be variant. Some of these might be common SNPs, some of them might be rare, some might be unique to you. But sequencing allows you to identify all the variants. Technologies such as exome, which is the sequencing of just the genes or the protein coding regions, or whole genome sequencing, are just beginning to become available to the general public. And as they become more readily available, that's going to cause an interesting change. At SelfDecode, we are in the process of preparing ourselves to incorporate this type of information, as well as improving on our genotyping chip data that you get from 23andMe and Ancestry.com. These scientific studies identify SNPs that are associated with all sorts of traits. So it could be height, it could be eye color, it could be anxiety, it could be diabetes. The identification of a SNP that he associates with a disease is termed a risk allele. So the risk allele refers to which of the two letters, is it G or is it T? So the allele, say T, is associated with diabetes, then T is the risk allele. So having this allele means there is an increased chance that you might have or will develop the disease. This information can be difficult to process, as the studies are all slightly different, and interpreting their meanings or their results can be pretty tricky at times. So at SelfDecode, we provide summaries of the information available for each SNP. And it's important to realize that not all SNPs have research out there about them. So you might find SNPs on SelfDecode without much information. And that's not because we're not giving it to you. We are very careful to get everything we can find. It's just that it's not out there yet. But more information will be coming forward over the years. So for example, this is SNP RS882632. The minor allele T occurs in 10% of the population. The major allele C occurs in 90%. My genotype is TT, and so I'm homozygous minor. And that occurs in 2% of the population, as you can see down here. We also provide a summary of the study that produced results with this SNP. So that's this bit here. So this study associated unipolar depression with this SNP. There's a couple of things I'd like to draw your attention to before we move on. The first is called the p-value. That's this one here, p equals. Now this is the probability of obtaining a different result. So the lower the p-value, the less likely it is that this is the wrong way. Okay, so it's a funny kind of reverse way to think about it. The easiest way to think about it is the lower the p-value, the more likely this SNP is associated with this trait. So you're looking for low p-values. P-values below, say, 0.0001, anything below that, so 1 times 10 to the negative 3 and below are getting good. The next one I want to draw your eye to is the impact score. So this is the degree of influence an allele has on a trait. So the p-value tells you, is this SNP associated with this trait? The impact score tells you how much this SNP actually affects the trait. Anything above 4 is a significant level of impact. If you hover over these little I buttons, this will give you a bit more information about them when you're using SelfDecode. The other thing I want to draw your attention to is the odds ratio. So this is whether the risk, or say the effect of a SNP increases or decreases. So if the odds ratio is above 1, then the risk of developing or having that trait is increased. Whereas if the odds ratio is below 1, then it's decreased. So just as a summary for this particular trait, for depression, there's a strong association with the SNP. The risk allele is T. I'm homozygous T. It's a pretty strong impact. Not the strongest I've seen, but like it's good. And the odds ratio tells me that having this SNP increases my chance of unipolar depression. Okay, that's what the data tells us. We also provide links. So these little R's are going to link you to the NCBI website that has the paper that we're talking about here, the genome-wide association study. And you can go there and look at the information yourself. That's totally cool. It's worth doing, but it can get a bit tricky at times. There's a little bit of a cheat sheet here that are just things I'd like you to consider when looking at scientific literature, and a good way to approach it. The first thing is to skim the abstract. So everything important should be in that first little block of text at the beginning of the paper. Well, you want to ask yourself, what question are they trying to address, and is it relevant to your question? Because if it's not, they're not going to help you out. Then look at the key figures to get a quick understanding of what the results were. And then it's worth skimming the methods, just in case there's something strange about them. And then look at their findings. Just the last word, beware of statistics. So a 10-fold increased risk can sound like a scary thing, but if the probability of a trait is 0.0001 and you've got a 10-fold increased risk, then the probability of that trait for you is still 0.0001. It's still highly unlikely. Our goal at SelfDecode is to go through all this for you, to identify the key information and present it as clear and understandable manner as possible. And we are constantly working on it and improving and getting feedback from you guys and trying to incorporate it in the website to make it a better and stronger website. Okay, let's just quickly summarize the SNP section, and then I'll move through to a couple of examples and then we'll go to questions. So we started with a sequence of DNA, and we recalled that we get a copy from mum and a copy from dad. We looked at how we write our genetic variation, whether we're homozygous or heterozygous for a genotype. We talked about alleles being G or T, whereas our genotype is GG or GT. We covered population frequencies, the databases that tell us how often genotypes are observed. And we looked at how we identify risk alleles and how we interpret scientific studies. And now I'm just going to run through some demos of the website and where you find this information. So here we are at the website, Decode Your Genome at the homepage. I'm going to go look at my SNPs. I'll start with my potentially bad SNPs. We'll have a look at those. So here's my potentially bad SNPs page. You can scroll down. You can see that each SNP has that RS and then the number that it describes it. So that's each of those is a unique SNP. If they're the same RS and number, it's the same SNP. And then we've got the gene with the information about the gene. And then it tells me my genotype. So the two copies, mum and dad both had an AA or both had a CC. And there's a bit of information about what the trait is and how it's associated. So these ones are all associated with different variations of disorders. And you can scroll down and have a look. Most of these appear to be just a single gene with a single SNP. So I want to find a gene with a few SNPs in it that might be able to tell us a bit more, be a bit more interesting. Okay, so this one here, FTO, we'll look at the first SNP. Let's open that in a new page. Okay, so here we are, SNP RS9939609, okay? Its parent gene is FTO. It doesn't mean it's actually in the gene, but that's the gene it's closest to. We've got the population frequency data. My genotype is quite low, but not super low. And it's associated with some different traits. So diabetes, increase in body mass index, more body mass index, increases hunger and caloric intake, associated with obesity. And then we've got some summaries of studies. So you can see the p-value, 1 times 10 to the negative 20, 1 times 10 to the negative 7. Impact's 20, so that's a significant impact. Again, 6.7, odds ratio 1.25, so this is increasing my chances of type 2 diabetes. Same again with the odds ratio. And then for each study that we've found that had information about the SNP, we try and summarize them in a separate box. And then here's our population frequency information. So all phase 3 individuals, this is all of the individuals combined. And then we've got different ethnic groups with different information. You can see that there's some differences between them sometimes. The Africans have almost a quarter, whereas it's low in other groups. Okay, we'll have a look at the gene, parent gene FTO. So we've identified that there's something wrong with my FTO gene, and I now want to have a look at what the gene does. It's involved in dioxygenase. And so a useful bit of information here might be the fixes. So you can go along and have a look at what things I could do to counter having a corrupted FTO gene. I could eat less, I could increase my physical activity, have curcumin, pomegranate, lower dietary protein, and there's more fixes that we can suggest. This is an aspect of SelfDecode that we're working really hard on, and we're trying to get more and more feedback to you about what you can do about having a gene that isn't working properly, or how to increase pathways that aren't quite there yet. Okay, so I'll just look at another SNP. This is RS4680. Again, it's in CompT. So this is quite a common one that people have. As you can see, 15% of the population have the genotype AA. And it's pretty evenly distributed amongst all populations. This is an example of quite a well annotated SNP. So we've spent time reading about it and putting all the information together in one place, so you've got a good idea of what the SNP does. It increases dopamine. My version increases dopamine in the neocortex, and it has some good and some bad consequences. It's the same with the warrior gene. We've got some good and bad consequences for the major allele as well. So these are the references. I'm just going to open some of these references in another page just so we can go have a look at them, just to give you an idea of what it looks like in PubMed. And here's some studies. This study probably isn't that useful. I mean, blood metabolite measurement doesn't tell you a lot, and it seems to be the same study twice. We'll be working at tidying these things up and giving you a little more information. So here's the PubMed, tells you about the SNP, the gene, and its involvement in gastric mucosa, and another study telling you about how it interacts with modafinil. So if you're using modafinil, it may be good for you, it may not. Okay. Just going to read through. There's a lot of good information. So it's mostly been summarized here, which is good. You don't need to spend time reading through all the scientific literature. We've got it all there for you already. We can look at COMT, what it does. There's the SNPs, there's my one there. And then we can also look at fixes. So fixes for low COMT. If you have low levels, what you can do. If you have high COMT levels, based on which of the two alleles you might be homozygous for. And just methods for increasing or decreasing dopamine. Okay, I'll give you one more example, and then we can move on to some questions that have been asked. So this is SNP RS1051730. It's in the gene CHRNA3, CHRNA3, I don't know. Not all genes have short names. Genotype A8, so it's a very rare one. Seems to be pretty prevalent in European populations, strangely, but not so much, say, East Asian or African. Okay. Yeah, that's weird. Right, we'll go back to the top. It is associated with traits such as nicotine dependence, lung carcinomas, smoking behaviors. Okay. So it affects my brain's ability to process nicotine, basically. Now, this is an interesting one because what the SNP is going to do is increase my likelihood of taking up smoking. Then because of that trait, it's also associated with a bunch of other secondary traits, such as lung cancer or chronic obstructive pulmonary disease. You've got to be careful to identify the primary trait that it's causing and then what that primary trait might then be doing later on down the line when you're looking at association studies. So the association might not be the direct association. It might be a follow-on effect from the primary effect of the SNP, or of the association. Sorry, I'm confusing myself here. Right, back to it. Yeah, these studies are all pretty good. I'll just have a quick look at the gene. Summary of you see, it's involved in neurotransmission. Yeah, so it's definitely a neurological or behavioral involved gene. We can go down. So we haven't got fixes for this one yet, but we've got ways to increase and decrease the gene. So if we want to increase it, tamoxifen looks like, so that double arrow means it can go up and down, whereas decrease the gene, of course nicotine's in there. Yeah, so this gives you some ideas of drugs you might want to take. Of course you should go talk to your doctor before taking any of these, but it gives you a bit more information to be talking to your doctor and be a bit more informed. You can also go to potential fixes. So this is where we list all of the fixes, and we can search for that gene in case I missed it. Okay, CHRNA3 isn't here, but CHRNA5 is. They're close associated genes. At the moment, our suggestion for fixing it is to not start smoking or use nicotine. Pretty good advice based on the likelihood that I'm going to become addicted if I do. Okay, so that's the end of my demonstrations, and we've still got 15 minutes. We had some specific questions that were sent in. I want to quickly, they're quite long and detailed questions, but so basically they're just two questions. So I'm going to go through these first because they're actually great questions. And then Martha hopefully will give me some questions that you guys have been asking during the talk, or if you've got any questions you want to be answered, put them up there now. Bear in mind, we've only got 15 minutes, so we won't be able to get through all the questions, but we'll do what we can. I'm sorry if I start talking faster towards the end, but I just want to get through as many questions as possible. So this one is starts with 3 questions. 1, what are the criteria that SelfDecode uses for choosing which SNPs to report for a gene? So our criteria is a little varied at the moment. We focus on SNPs that have information from studies, from GWAS studies or sequencing studies, but we also will include SNPs that have information about population frequency, because that can be useful. Often very low population frequencies is an indicator that a SNP might be bad, or it might just be rare. It's definitely not the reason. I wouldn't use a low population frequency, but yeah, we use a bunch of different criteria to help us choose which SNPs to report for a gene. What conclusions can be drawn from the mere fact that a SNP is included in the SelfDecode report? By SelfDecode report, I'm assuming we've identified it and shown it to you. I'd say it's not, if it's included in a report, it's not definitely going to affect your health or your well-being in any way. It's just that we have information on it and we'd like to share that with you to allow you to become a little more informed about it. So in the future, something we're working on now is creating a nice user-friendly interface that helps people see quickly whether or not a SNP has a particular impact or influence, or whether or not it's just something we have information about. So that's something we're working on. What conclusions can be drawn from a heterozygous or homozygous result for a reported SNP? Okay, first thing, you want to go look at the information, because sometimes there's actually information about this. It's a second thing, this is sort of going back to Genetics 101, something I didn't cover, but maybe I should have, and that's recessive and dominant traits. So a SNP might be associated with a dominant trait, in which case heterozygous and homozygous would be the same. If it's recessive, then heterozygous wouldn't show the trait while homozygous would. And you can have additive. So heterozygous might increase your chance 10%, whereas a homozygous having 2 copies might increase your chance 20%. It's difficult information to gather, and it's difficult. So the studies don't always include this. Where we can find the information, we include it, but it's not always known. What conclusions can be drawn? I'd say read through the information. Heterozygous, homozygous, having 1 copy, if it's a strong impact, is probably still going to have an impact. Yeah, okay. Okay, I'll read the next section. Let's pose a hypothetical situation in which 8 SNPs are reported for a gene. Okay. Obviously, if one of those SNPs has been widely studied, there'll be lots of references in the explanation of that SNP, and it should be given due accord. Yep, cool, that's what we're talking about. But suppose that none of these SNPs have any type of substantive studies associated with them, or that none of the associated studies are relevant to my concerns in a specific metabolic process. I'm left with a lot of uncertainty. Further, suppose that 8 of these 8 SNPs are heterozygous. Can I make a statistically based assumption that with this many heterozygous SNPs, the gene is probably impaired at some notable level? If so, what can I conclude if only 4 of the 8 are heterozygous? Okay, for starters, if there are no associated studies, if there's no information saying that these SNPs do anything, then I wouldn't assume that they do anything. If I had to make an assumption about a SNP, especially one from SelfDecode or Ancestry.com, and there isn't any information about it, I would assume that that SNP actually doesn't change anything significant in your body. It's just a SNP that you have that other people have a different version of. So if you've got 8 SNPs with no information in a gene, then I wouldn't be too concerned about that gene. Yeah, with the heterozygous homozygous thing, again, it comes down to information you can find from the studies. But yeah, if there's no information about the SNP, then I wouldn't worry about it too much in that gene.
Can I add to this one?
Yeah.
Okay, so if there are studies that associate the SNP with other diseases, right? So you can probably assume that the SNPs lead to some kind of a malfunction of the gene, or it's associated with their malfunction of the gene. So in that case, even if it's not directly related to the disease or the metabolic process that you're looking at, I would suspect that there might be an issue with that particular gene.
Yeah, I'd read about that other process and learn about and see if it's relevant to me.
Yeah, and then I'm a geneticist. I would also look at their animal studies as well. But right now, most of the studies we have are human studies. So yeah, but just when we write self-hack, we look at their animal studies and see if there's any crossover or related information with respect to their processes. So I guess eventually we'll get there.
Yeah, we're definitely working on all those. Okay, so there was a second part to this question. In order to move beyond specific SNPs and to focus on the whole system, which is something we're very much interested in doing as well, we're working on it. I've attempted to transfer results for specific genes to a simple highlighting of genes in pathway diagrams, such as methylation. So pathway diagrams are very helpful. The gene has a SNP that is known to functionally impair that gene. I highlight the gene with a bold outline and note the major SNP. You also highlight genes with a number of homozygous or heterozygous results with little information. So again, it's the same thing, I guess. I wouldn't assume that just because a gene has a lot of SNPs, that gene isn't functioning properly. Often the SNPs aren't in the gene, they're located near it. If there's no information about the SNP, then it's probably not causing any major problem that has been looked at in a scientific study yet. But, keep your eyes open. Maybe in the future someone finds information about the SNP. But right now, if there's no information about it, don't assume it's bad. Specific question number 2. 2 years ago, my ND doctor recommended I have a gene test, which was done by Genoma Diagnostics. She requested their neurogenomic profile from buccal cells. I'm 64 years old and I have struggled with anxiety and poor stress response for many years. My HPA axis and circadian rhythm are out of balance. Heterozygous for MTHFR. Yep, okay. And COMT, yep, those are pretty common, also very interesting variants. GSTP1, so 2 variants in GSTP1 and SOD2. All right, okay. My doctor also thinks I have an MAO gene mutation based on symptoms. I recently listened to a what's the question? I have a SNP in my FK clock genes, okay, possibly based on circadian stuff. The HPA axis again. My doctor doesn't think I need to do any further gene testing and feels treatments can be provided based on symptoms only. I don't agree with that. I'm not sure I agree with that. Yeah, thank you. Yep, we're on the same page. It seems like the higher my knowledge level, the better I can control my current and future quality of life. Exactly. That is exactly what we're hoping to do. Okay, maybe there's more. Yeah, okay. 2 questions. Out of 22,000 genes, how many of these genes can be accurately tested, and how does one determine which of the testable genes should be tested? Okay, so of the 22,000 genes in our genome, so 23andMe has 602,000 SNPs that it's looking at. So, and they try and place them close to genes or within genes. So of the 22,000 genes that you have, most of them will have a few SNPs associated with them. So a 23andMe test will look at, say, 12 single nucleotides near a gene, per gene. That's not going to give you information about the actual gene itself and your own unique genetic variation. It just tells you common polymorphisms. Which of the testable genes should be tested? It's a harder question. I would look at your symptoms, and then, I mean, Joe's really good at this. He can listen to someone's symptoms, and then he's quite quick at coming up with genes that are of interest. My specialty is autism, so I'd probably, if you told me about someone's autistic symptoms, I might be able to give you some interesting genes. But yeah, so you can do some research about your symptoms and look at which genes. Another option would be to just sequence everything. I mean, a gene test, sequencing, say, 20 genes isn't that much different in cost to sequencing all of them nowadays. It's a whole brave new world. Okay, question number 2. can genes be turned on and off through environmental and lifestyle factors? Yes. If so, to what extent? Yes, that's the thing. So genes, every cell has different genes turned on and off. You don't have all of them on at once. You don't have them all off at once. You can take different supplements. So resveratrol is a classic molecule that turns on genes. It turns on your sirtuin genes, and they promote cell repair. They help your cells stay healthy for longer. They promote long life. So that's an example of an environmental or lifestyle factor that regulates or influences the regulation of your gene. So it sounds like there's a lot we can do to upregulate our gene pathways. Yep. But how can this be done if there are additional gene SNPs that have not been identified for the patient? Okay, so maybe this is talking about that whole SNPs outside of what the genotype chipping is looking at.
Looks like she hasn't even tested these genes, or she doesn't know her genotype.
Yeah. If you don't know your genotype, then I wouldn't start just turning genes on and off just based on symptoms. You want to look at least get a 23andMe and Ancestry.com. If you want to get a better, that'll give you a starting point for understanding what genes need to be treated.
Right.
Yeah, okay.
And also for the circadian rhythm and HPA axis dysfunction, there's a lot of environmental factors, right? That's why a lot of people will feel completely fine if they live a poor lifestyle when they are younger, and then as they get old, they start to feel it, right? Yeah, so some of it's in the genetics, but a lot of it is also in the environment.
Yeah, so, genetics is only, it's a tool that you put in your tool belt and you use with all the other tools to build your healthy house. It's one part of it. It's just that we've never had access to it like this before, but it's not just genetics and it's not just environment. It's nature via nurture. They work together to help create a healthy you.
Right.
I think that's the last of the questions that were pre-emailed, so we can...
Okay. So someone asked about the genes for autism and Wilson's disease. So I don't know if you feel like you need another webinar to go through autism genes, or you can answer the question here.
I can answer it, but not answer it. So basically the short answer is autism is a really complex disorder. And there's a lot of genes. They think thousands of genes that contribute to autism. There's three main groups. There's genes that influence the way that your synapses and neurons connect with each other. So there's structural genes. There's genes that influence the regulation of other genes during development. So a mutation in that gene might mean that during the fetus's brain development, it doesn't quite follow the normal pattern. And there's genes involved in the recycling or the sort of autophagic processes, so the recycling of proteins in the cell, which lead to problems in the neuron and the immune system, which is why a lot of people with autism have problems with their gut and their immune system. But yeah, there's a lot of genes involved. Okay, any other questions that we want to go through?
So do you know how they decide their score for importance of the SNPs?
How do we assign the importance of the score of the SNPs? This is something at the moment I'm not too sure about. So I've only just got on SelfDecode and I've been talking to Joe and Dominic about it. They've got an algorithm that will look at the p-value and the impact of taken from different studies, and they kind of try and combine those different types of information to come up with a single score. It's really complicated stuff, and to simplify it, you got to be careful that you're not losing the information by simplifying it too much. But having said that, yeah, I think it's based on the p-value, so how confidently associated the SNP is with the trait, the impact factors, so how much the SNP influences the amount of the trait, and then maybe the odds ratio, which tells you whether it's influencing it in a positive or negative direction.
Mm-hmm.
Yeah.
Okay. And are you done with that? answer?
Yeah.
Okay.
Hopefully.
Let me ask, do you indicate whether a SNP is a down regulation or an up regulation?
Yeah, okay, so that's the odds ratio value that you can see in the summaries of the research. You look at a SNP in SelfDecode, and if it has information about research that's been done on it, then you will be able to see if it upregulates or downregulates that gene. Again, it's not always known as well. Sometimes we just, because it's an association, all we know is having that SNP is associated with this trait, but we don't know what's happening in between that. We don't know how the SNP or what mechanism is occurring between the two, so we can't provide that information at all times. Let's see if we can find one. But yeah, any other questions?
So Debbie asked, will you be adding in a search module for pathogenic ClinVar SNPs? So I guess ClinVar stands for clinical variations.
Yeah, I know ClinVar.
Okay.
Yeah, that would be a great tool to include, definitely. I think there might be, so we've looked at ClinVar and we take into consideration their view on the SNPs. And I think it definitely, that feeds into how we weight and rate and give them a score. I'm not entirely sure though, I'd have to talk to others about that. But yeah, definitely we will be using, or we'll incorporate something similar to ClinVar that tells you which ones are pathogenic and have been identified.
Okay, so we should totally do that, right? Okay, so there are a few webinar topics suggested in the chat box. So we're gonna save this and plan the future webinar.
Yeah, more webinars.
Satisfy your curiosity here. So Debbie asks, do you do one-on-one consultations?
Hey.
So right now, I and Joe do consultations. You can look at selfhack.com/contact or selfhack.com/consult -specialist to see about our consultation services. So if you would like to consult Brendan, you can email us as well, I think.
Yeah, so any consultation I do would be through SelfHack, SelfDecode. So yeah, go through those guys.
I don't think you have a consult page per se, right? But
No.
I mean, yeah, if you want to consult Brendan, just email support@selfdecode.com and we'll hook you guys up. So yeah, but I mean, these are not medical advice, and we are academic geneticists. There's a disclaimer.
Yeah, instead of consult, maybe conversation.
Okay. All right. Well, I think that's the end of it. Thank you so much, everyone, for joining us tonight.
Yeah, thanks everybody for listening to my 101, Genetics 101 talk. Sorry if it was a bit basic for some of you, but kind of want to get everyone on the same page. A few
right.
future talks we can cover more interesting topics.
Thank you, William.
All right, cheers guys.